Welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson, and today's topic is DNA structure. Let's learn together. All right, so today we are talking about DNA structure, and we're also going to touch on the origin of DNA as a molecule. So we have to first start with what is it? We've talked about it in previous videos, and you've heard me say that DNA is our genetic material and that it stands for deoxyribonucleic acid. But DNA is really our universal genetic code. It's like the language of our cells. It's what our cells speak. It's what they use to create things. It's what they use to make proteins. It's what they use to do work. And every single cell on Earth can read and write DNA. And although all cells use the same language, every single organism is unique. We're not the same as a banana or a fruit fly or a dog, right? So even though we have this same DNA language, the order that our DNA is in is different. And that's what creates our own genetic story. So it'd be like if you wanted to read your biology textbook for fun. You might, I don't know. I wouldn't, even though I'm a biology teacher. I would prefer to read something like Harry Potter for fun. These are completely written in English, both of these books, but they're for very different reasons. They serve a different purpose, and they have very different stories. One's academic and one is fun. Not to say biology is not fun, you know what I mean. So that's the same thing with DNA. DNA is that base language that cells can use. So where did it come from? It didn't just poof into existence one day. The world just doesn't really work like that. So DNA, as we're finding now, and this has been research that's been coming out in the last decade or so, DNA has been found to be modified RNA. And RNA is ribonucleic acid. You should be able to say, okay, there's probably some similarities here. And DNA sugar deoxyribose is actually a reduced version of the RNA sugar ribose. So ribose is a pretty normal sugar. Deoxyribose is a little bit rarer. It's not found as commonly. So the scientists kind of hypothesized that DNA most likely came from RNA because it was a more simple version. And as far as we know, that's the story. Right now, this is a theory. We've been testing it over and over again. But remember, theories in science, while they are the best we have so far, they're not 100%. This is still potentially in that hypothesis range. So it hasn't been tested hundreds or thousands of times. It's not something that's 100% accepted. But as far as we know, this is what the story is so far. And again, we've got some similarities between our DNA and RNA, which we will go over in another lecture later. But we've got a couple of strands here, which are going to be our backbone containing our sugar and our phosphates. And in the middle, we've got some bases. For RNA, it's the same thing. You see a backbone and you see some bases hanging off of it. So very similar, but slightly different. Now let's get into that structure of DNA. I said some words that we probably haven't heard before, and I'll go over those again. DNA is deoxyribonucleic acid. Say it with me. Deoxyribonucleic acid. It's a fun one. Pull it out at parties, say it to your friends, amaze and astound them. It's a good time. The D is deoxyribose, the N is nucleic, and the A is acid. That's where we get that acronym. It's a polymer. So remember, polymers are big structure, and it's made of these nucleotide monomers. Monomers are those little building blocks. And there are multiple parts to a nucleotide. We have a phosphate, a sugar, and a base. That sugar is called deoxyribose. That's why it's DNA. If you're thinking, okay, well, why is RNA called RNA? The sugar is ribose with an R. So that would be one of the differences. We're going to focus more on these bases in a moment. We have 4 letters in this alphabet. So think back to where we saw that biology textbook and Harry Potter, that other book. Those are made of 26 letters in English. We only have 26 letters, but it creates something so vastly different. So all the literature, all the books, all the magazines, all the blog posts you've read, they're all made of the same 26 letters in English, if you're reading in English. And in DNA, we only have 4 bases. So how do we get so complex with life and then keep it simple? Here's why. We've got adenine, thymine, guanine, and cytosine. I've heard people say guanine. It kind of makes me feel weird, like I'm saying gooey. So I say guanine. there you have it. These are our 4 bases. And you'll notice they all have a phosphate and a sugar, but then they differ in that they may have 2 rings here, like guanine and adenine, or they could have a single ring, like cytosine and thymine. That would be really wide if you need different numbers to fit together. So I'll show you that real quick. Got excited with the animations, guys. It was fun. So A always pairs with T and G always pairs with C. When you look at it across this way, they're going to fill 1, 2, 3 rings when they pair up, and 1, 2, 3 rings when they pair up. These are not paired together yet. They are not bonded. So this is not how it would look in a DNA molecule yet. But that's how we always pair it. The way I remember this is apple is in a tree and a car is in a garage. Now, I am from Texas, so I also say the name of the city I'm from. You could say Abilene if you want, whatever city. Abilene, Texas, go Cowboys. If you don't know anything about football, that is okay. Just think apple in the tree, car in the garage. Much simpler. Now these do form hydrogen bonds with each other, and for some reason it's not lining up. So when we think about how we need these to fit together into a DNA molecule, they're going to kind of organize themselves a little bit like this. We have our adenine and thymine, cytosine and guanine, and they're going to pair up in a very specific way. Adenine and thymine are going to pair together. Guanine and cytosine are going to pair together. And this happens essentially because DNA has to be the same width all the way across. We can see here that when you pair up these 2 different types of base pairs, you get 1, 2, 3 rings going across. If you were to pair up some of the other combinations, you would get different combinations or a different width all the way across. If I tried to pair up T and C, for example, I'd only have 2, and our DNA molecule would do this. It would go in at points and out at points, and the same is true of G and A. There would be too many, it'd be too wide to be the right distance across. In addition, there are different numbers of bonds between the 2. Thymine and adenine only have 2 bonds. Cytosine and guanine have 3 bonds going across and connecting them. So you couldn't have a T and a G matching together because they'd have the wrong amount of bonds, and the DNA molecule wouldn't be sturdy enough. The way I remember these pairings, apple is in a tree, a car is in a garage. That's the best way I can think of to remember it. Apple in a tree, car in a garage, besides just memorizing that they go together. Because I'm from Texas, we like to put a city name here and then use Texas. So it would be like Abilene, Texas, go Cowboys, who are obviously one of the Texas football teams. So that would be my example. Now they do form these bonds, and they're hydrogen bonds, which are really, really important because if you don't have those hydrogen bonds in the middle of your molecule, your DNA is disconnected and it wouldn't stay together like it's supposed to. They form this backbone here, so we have a sugar phosphate backbone, and it's going to make almost like a ladder going all the way down. And you can see that here if we add in another level, another apple in the tree. We've got this other base pair here. It's going to be connected to this backbone, just like that. And so on and so forth. I know that may be hard to see, but it's the same as you see up here, just flip-flopped with the A and the T. So, getting into a little bit of history here. The shape and structure of DNA was not discovered that long ago. Your grandparents were probably not taught about DNA in school, which is a little crazy to think about. It was discovered by 2 scientists, Watson and Crick, in the 1950s. I say discovered, they put all the different information together. They took a lot of different people's information and tests and labs and put them together to create what we know now as DNA. But there were other scientists that worked on it as well. What they found was that bases form the steps of a spiral staircase. But the sugars and the phosphates are going to form the edges. They call them handholds. I don't get handholds. I just think of it the sides of the ladder. A little bit simpler in my brain. And then twists to form a double helix. So it's a spiral structure, and that's why they call it a spiral staircase. The other scientists that they got a lot of this information from, and a lot of it about the actual shape of DNA, the double helix, was from a female scientist named Rosalind Franklin. And she unfortunately died before a lot of this was put together, but they took her images and did not give her credit, which unfortunately used to happen quite a bit in the scientific community. So we call it a spiral staircase. You can see here an actual spiral staircase. And you can see that the shape is very similar between DNA and that staircase. In the middle here we have the rungs, or what you put your feet on. And then on the outsides over here, you'd have the railings, for lack of a better term. Chargaff is another scientist, and he had a rule that he came up with to figure out how much of each base was in a DNA molecule. The amount of adenine in a DNA molecule was equal to the amount of thymine. Chargaff essentially figured out what base pairs went with which. The amount of guanine also equals the amount of cytosine. This should make sense to us. They always pair up, so they're always going to be equal amounts. You're never going to have a C and a T pairing together because the bonding and the width will not make sense. So here's an example. If a DNA molecule is 10% adenine, how much would be cytosine? This is a little bit of a math problem. So let's kind of break it down. We have adenine and thymine, and we know they have to be the same. So that's going to be 10% here for thymine and 10% here for adenine. So that is a total of 10 plus 10%, 20%. Now you can only have 100% of something. I know we give 105% if we're out on the field playing football or on the court playing basketball, but you only have 100%. So 100% minus that 20, the 10 from adenine and the 10 from thymine, gives us 80% left over. We know we have to have cytosine and guanine, and they're going to make up our 80%. So if they're equal, we're going to have 40 and 40. Let's check our math. We have 10% adenine, which means we have to have 10% of thymine. That is going to be 20%. That means we have 80% left to kind of wiggle with here. 80%. Now if guanine and cytosine have to be equal, 80 divided by 2 is 40. And that's how we figure out how much percent of each of the bases there are going to be in a DNA molecule. Now, no matter where you are, where you're from, what you look like, you are 99.9% the same as every other human being. Again, this is an average number of base pairs. It's from the Human Genome Research Institute, so I like a lot of their stuff. It's pretty cool. So you're about that much the same as everybody else. Now I want to compare us to a couple of other organisms so that we can take a look at the differences and the percent that we have in common with some other organisms, and they may surprise you. That 0.01% is what makes us different. So that very, very tiny amount is why you see different hair color, eye color, heights. You see different facial features, different skin color, things like that. That's in that 0.01%. So we're way more similar than we are different. If we take a look at eye color and the DNA code for it, we'll see that it starts out all the same, GAT. Those are all bases. Now we'll notice here, green and hazel eyes continue to be the same until we get here. Then there's one base different. Brown eyes, they change pretty quickly. Instead of having a CTT like green and hazel, it's CTA. So they do have additional base here that's different. They also have another base over here. Blue eyes are the most different. They're the same for that first triplet, but then they become CGC, then CTA, and so on. So it's these bases that are slightly different that give us all those different eye colors, which is pretty cool. We share about 96% of our DNA with a chimpanzee. This should make sense. They have all the same body structures as us, but we're bigger, we stand a little bit taller, we have some different facial features and so on, but all of our internal components are the same. We share about 90% genetic similarity with a cat. Again, they're still a mammal, so they have organs like we do, they have organ systems that are all the same as ours, but it's still about 90% the same. Between us and a mouse is about 85%, so we should feel like we're getting a little further away from a human here, even though mice are still mammals. They have live young, they have all the same organ systems we do, but obviously a mouse and a human are a little bit more different. Similarity between us and a cow is about 80%. Us and a fruit fly share about 61% of the same genetic information, which is kind of crazy to me because we are nothing like a fruit fly. We share less in common with a chicken, which again should surprise some of you. We share less between us and a banana. Again, remember, bananas have DNA because they are living things. I get asked that question every time. The last thing I want to talk about today is the universality of DNA, and really want to make this point. We can transfer genetic information between species. So just because there's a gene in a pig or in a cat, or in this case, in a jellyfish, it can be transferred because DNA is all the same, just in different orders. So there's something called GFP, and that is the green fluorescent protein, and it's found in certain species of jellyfish. And it's what makes them glow green, like you see in this picture. This is an actual picture, it's not fake. When they take that gene out of the jellyfish, they can isolate the gene, and then they can put it into different embryos or developing babies of either a pig, a cat, mice, or a monkey. Those animals will also glow green. So their cells will glow green. And that's what we see here. So it's a protein from one, or a genetic combination of DNA from one species, and it can be put into other species. So DNA is always the same. The reason we look different is that it's in different orders. Let's recap what we've learned today. We learned about the origin of DNA, and that DNA came from RNA. So RNA was around millions of years ago and slowly evolved over time to become what DNA is today. In addition, we learned that DNA is the language of cells, and it basically has all the instructions for what your cells need to do and create. It's made of monomers that are called nucleotides, and each nucleotide has 3 parts: a sugar, deoxyribose, a phosphate, and a base. And these nitrogenous bases can be one of 4: adenine, thymine, cytosine, and guanine. Adenine and thymine always pair together, and they have a double hydrogen bond. Cytosine and guanine always pair together as well, and they have a triple hydrogen bond. Please like and subscribe for more biology videos. I really enjoyed having you here today. I hope you had fun. I hope you learned something, and I'll see you later.